Okay, so the type of bond that you specifically asked about were polar covalent bonds. We have some other types of bonds that we want to know what they do with water as well. So there's polar covalent bonds. Remember, there's also non-polar covalent, covalent bonds. And the last one that we wanted to know what happens with water is those ionic bonds. So let's start with our two types of bonds that have this word covalent in them. Can one of you guys help me out? When I use the word covalent to describe a bond, what happens when we have a covalent bond? What kind of bonds are covalent?
If they share electrons.
Absolutely. Yep, that's absolutely correct. So whenever I use that word covalent, it always means we're doing, I like to call them a sharing bond. We're always sharing our electrons. And that's something that popped up on the homework. I'm sure you guys noticed it. It's always electrons that we're sharing. We're never sharing protons, right? We're always sharing those electrons. So we've got two types of sharing bonds going on here. We've got what are called the polar covalent bonds and the nonpolar covalent bonds. When I talk about polar covalent compared to nonpolar covalent, the difference between them is going to be how they share those electrons. So is it those polar covalent bonds or the nonpolar covalent bonds that they share the electrons equally? Who does equal sharing? Do we remember?
Nonpolar.
Nonpolar, yes, absolutely. My nonpolar covalent bonds, nonpolar is going to tell me that we do equal sharing, which means that this polar word over here, polar means I am doing unequal sharing. I kind of like to joke, I have a 3-year-old at home. So if we do any sharing, which we're still working on, we're very much polar covalent sharing over here because she's not quite good at 50-50 splitting yet. So polar covalent bond is what toddlers do. We might share a little bit, but it's definitely not equal. Non-polar covalent bonds, we are sharing equally, sharing those electrons. That's completely different from this other type of bonds that we have over here called ionic bonds. When I build an ionic bond, what am I doing with those electrons in an ionic bond? We're not sharing them anymore. What's happening in an ionic bond?
Transferring electrons.
Yeah, absolutely. So an ionic bond, the nice way of putting it is that we're transferring electrons. But a lot of times I actually like to use a little bit more of a blunt or a ruder word here. I like to say that we're stealing. This is a stealing bond. So either, to put it nicely, I've transferred my electron to you, or to be more blunt, you've probably stolen it from me because I got close enough to you and you say, oh, I really want that electron, and you just took it from me. So when we have an ionic bond, that's not a sharing bond. That's a transferring bond. We move an electron from one place to another. When we talk about covalent bonds, these are always going to be sharing bonds. Now, the reason we were talking about our types of bonds, especially polar covalent, that's the big one that we were asking questions about. The reason we're talking about the bond types is because big picture, what we want to know is what happens to these bonds in water. That's kind of our goal. We want to figure out what's going on with each of these when I put a compound that has them in water. Now, the best way to think about what's happening with these is to kind of think of some examples. So let's start with an easy example of something that's built with an ionic bond. When we talk about ionic bonds, the compound that we talked about a lot with an ionic bond is salt, right? We use sodium chloride. Sodium chloride is a molecule that's made with ionic bonds. Let's do a little reminding for ourselves here. With an ionic bond, we're transferring those electrons. The type of bond that I form comes down to what's called the electronegativity of the atoms that are inside of it. Can someone help me out? When I use that word electronegativity, what does that mean when we're talking about electronegativities of atoms? How could we describe that?
How strongly they hold electrons to itself.
Awesome. Yeah, it's how strongly they hold on to things, right? Or how badly they want another electron. We're not really going into the specifics of why. When you take your chemistry class, they'll explain to you why some atoms want electrons really badly and some don't. So electronegativity is it's a measure of how badly an atom wants electrons. Electronegativity matters because it's going to tell me, are you going to be nice and do a sharing bond? Or are you going to be mean and do a stealing bond? Are you going to take electrons from me, or are you going to share electrons with me? When we talk about an ionic bond, we are talking about things that, help me out here, their electronegativities are close to each other or they're far away from each other. If I steal electrons from you, are we closely matched or not closely matched? I want to punt the question to see if Elaine or Maria has a guess for me. If we do an ionic bond, do you think they have matching electronegativities or very different electronegativities?
Different.
Yeah, very different. Absolutely. Yes. So when I form an ionic bond, we have very different electronegativities. This is going to make it happen where I steal an electron from you, because somebody, in this case chlorine, when we talk about salt, chlorine really wants that electron. Sodium, not so much. It doesn't care as much. So chlorine will actually steal the electron from sodium. The reason that matters is because when we're talking about what's happening to our bonds, when I put them in water, it's going to matter to us that I stole an electron. Or maybe to put it another way, it's going to matter to us that we're not sharing our electrons. If we just transferred electrons from one atom to another, when I put those atoms in water, when I put salt in water, remember that salt completely dissolves. So we'll say that sodium chloride dissolves, can't spell, dissolves in water. The reason it dissolves is because sodium has a positive charge. Hey, I bet we can answer this question. Sodium has a positive charge. That means that it's a type of ion. Is it called a cation or an anion? Since it has a positive charge. What's the name of those positive ones?
Cation.
Yeah, the cations. Absolutely. Yep. So sodium, because it's a cation, has a positive charge to it, sodium is going to get attracted to oxygen's negative charge, because oxygen has a negative dipole, is what it's technically called. So let's add that on there. Oxygen's negative dipole. And chloride, which is my one with a negative charge, the one that got the electron, that's called an anion. That's going to be attracted to hydrogen's positive dipole. And remember, the only thing, we said in our guided lesson, the only thing that holds an ionic bond together, that keeps these two things close to each other, is that they have an opposite charge. So if I bring in something else that has charges, then I have no reason to stay together anymore, because I found another charge that makes me just as happy. So ionic bonds, I think we kind of equated them to kind of like puppy love, right? To high schoolers, that they have opposite charges. They're so attracted to each other, but bring in something else with a charge and it probably won't stick together. When we start talking about covalent bonds, when we start talking about our sharing bonds, it doesn't matter if you share equally or if you share unequally. The fact that you're sharing means that you will not dissolve in water. So that's that's a big picture idea here. Let me put that. I'm gonna put that in red. We're gonna say any. Oops. Let me type here. Any atoms that share electrons with each other. I should have said molecules. Oh well. Do not dissolve in water. If we were in class together, if we were doing a regular lecture, I like to use this phrase. This is an underline, highlight, star idea. Like whatever you do in your notes to tell you it's important, here's something that we would want to flag as important. If I'm sharing electrons, it doesn't matter if I share them equally or unequally. I have this level of commitment where we're stuck together because we're sharing our electrons with each other. So we're going to share our electrons with each other, which means that you can put me in water, but I'm not going to dissolve, or I'm not gonna detach from my other atom that I'm sharing with. We're gonna stay stuck together. Now, polar versus nonpolar, they're gonna do slightly different things when we put them in water. Does anybody remember for me, what was the example of the nonpolar compounds that we had in the notes? What did I tell you was nonpolar? Or maybe maybe let's put it another way. Do we have an idea of what happens when we put nonpolar things in water? Do nonpolar things like to be around water?
No, it doesn't mix.
Yeah, they don't mix together. Nonpolar things don't like water, so that's a note for us. Nonpolar, ooh, we're still caps lock. Nonpolar molecules don't like water. The example we gave of a nonpolar molecule was oil. If you look back in your notes, kind of toward the end of, I think it's the end of lesson number 3, there's the picture of pouring some oil on top of water and how it forms that layer on the top and the water's underneath. Whenever I do something that's made with a non-polar covalent bond, non-polar things in general don't play nice with water, is the way we described it. Or to put it in another word that we used, we called non-polar things hydrophobic. They're afraid of water. So non-polar things, we are sharing electrons, which means number one, we know we're not going to dissolve. There's no way we're going to dissolve. But number two, the fact that we share electrons equally in a nonpolar bond means I want nothing to do with water. So I'm actually going to squeeze the water molecules out. We're not even going to mix. So we could say they don't like water, and nonpolar molecules don't mix with water. They'll form that layer on the top of water like we saw in that picture. It's a little bit more complicated when we talk about polar covalent bonds, because when we talk about polar covalent bonds, these are not sharing their electrons equally. When we don't share electrons equally, we form a structure called dipoles. So hopefully that word sounds a little familiar to us. Dipoles. That's when somebody gets the electrons more and somebody gets them a little bit less. So unequal sharing with our polar covalent bonds means that I have dipoles, positive and negative charges. Mix me with water, I'm still going to stay attached to the atom that I'm sharing my electrons with, because we're sharing, we're connected to each other. But at the same time, I'm not afraid of water anymore, because water, here, let's draw, I'm going to try to draw here. It's going to look real terrible. Here we go. I'm going to draw a little water molecule. Remember that water, H2O, has areas that are partially negative. Here's my partially negative area. And I've got areas that are partially positive. When I put something like this with a partial negative and my partial positives next to something else, Up here, remember that this kind of bond up here, the nonpolar covalent bonds, they were saying, I don't need your negativity in my life. We're equally sharing our electrons. We're happy. But something over here that's not sharing equally, if you've got a partial negative and I've got a partial positive, I'm kind of attracted to you. I might want to form a hydrogen bond with you. Or you've got a partial positive, I've got a partial negative, I'm kind of attracted to you. We might form a little bond together. So when we talk about a polar covalent bond or a molecule that is a polar molecule, the first thing for us to know with it is it's not going to dissolve in water. It's always going to stay attached to who it was already attached to. But the second thing that we should know about it is it will surround itself by water. So let me write that down for us. Polar molecules will surround themselves with water. So we'll draw ourselves here. Got a little sugar molecule. Oops, drawing right here. Here's a little sugar, because sugar is an example of a polar molecule. What'll end up happening with that sugar is we'll put water all around it on all of its sides, because sugar has some places that are partially positive and some places that are partially negative. But what's not going to happen is I'm not going to chop this sugar molecule in half like we did over here with the salt. Remember that salt completely falls apart. That bond completely breaks. Over here with sugar, with a polar covalent bond, we keep those bonds intact. Sugar is never going to fall apart, but it's going to completely surround itself by water molecules. So when I put something with a polar covalent bond in water, it will play nice with water, but it's not going to dissolve in water. It's not going to fall apart in water. And the reason it won't fall apart is because we are sharing our electrons. If we weren't sharing, if we were just stealing or transferring them, we're going to fall apart. You put in other charges, we're just going to completely fall apart. But anytime there's a sharing bond, anytime it's covalent, we're still going to stay attached to each other, even if you put me in water. I know that was a question that you brought, Maria. What other questions? Does this make more sense now? Or what follow-up questions do we have about our types of bonds?
Oh, no, thank you. Now I'm more clear with this information.
Good. Yeah, that's kind of our big picture with the bonds is if you're sharing, you're in a committed relationship. You're not going to fall apart. If you're stealing or transferring, yeah, you're probably going to fall apart. It's just going to happen. So that's maybe the simplest way to remember what happens in water with those. Jalen, did you have any particular questions today?
I didn't have any specific questions. I just kind of wanted to listen and see if I could.
Yeah, absolutely. Or if there's any, and I'll toss this out there for everyone, Maria too, if there's any other topics we wanted to talk about today, because we got time. Otherwise, we can all kind of stare at each other.
Okay, no, if you could like just explain to me a little bit, because I get a little confused about when you talk about plasma, because you have the cytosol. I'm clear that the cytosol is the liquid that's inside the cell, and extracellular is the one that's outside of the cell, and the plasma.
Okay.
And the plasma never get mixed with cytosol or extracellular. No, it's something different.
Yeah, so this actually would be a great thing for us to talk about too, our types of fluid. So here, let's make a new board. We are interested in cytosol. We are interested in plasma. Trying to think some of the other ones we talked about. We had that one with the big long name, right? Interstitial fluid. And then the other one that we mentioned that I'll put on here is called lymph. These are the big ones that we talked about. I think it was just those three big ones, then we had some others. So you did use some terminology that was perfect. You used the term extracellular fluid. That is in comparison to what we call intracellular fluid, right? The reason I put my label over here kind of in the middle is we actually have a ton of different kinds of extracellular fluid, like you know from your studying. So the one that we're interested in talking about first maybe was the plasma. But the one that we actually have the most of in the body is here our interstitial fluid. We don't have a ton of lymph, but that's kind of the next most abundant that we have in the body, and then a whole lot of other little ones. The thing with extracellular fluid is that technically all of these kinds of fluid here, they're all in kind of the same circle of life, meaning that the only difference between these fluids is the location that I find them. So let's start here with the plasma. When we talk about plasma in your body, where would we say the location, the specific location of plasma is? Where do you find plasma?
Inside the blood vessels.
Yeah, so plasma is the fluid inside blood vessels. That's where plasma in particular is found. Plasma is an extracellular fluid, so it's inside the blood vessels, but it's outside of the red blood cells that live inside those blood vessels. The way I know it's outside is because it's one of my extracellular fluids. It's one of those outside ones. So plasma is what I call the fluid that I find inside your blood vessels. If you donate your blood, they can actually spin down the tube and get all those red blood cells at the very bottom and see your plasma on the very top of it. So first kind of fluid is the fluid that I find inside those blood vessels. Your blood vessels, especially when we were talking about our types of blood vessels in the body, remember we had the ones that were called the sinusoids that are really leaky. This plasma, this fluid can actually leave the blood vessels. And when it leaves the blood vessels, now it gets a new name. Now I'm going to call it interstitial fluid. So when I talk about interstitial fluid, this is going to be the fluid that is inside tissues. And this is a term that's going to matter a lot more in lab this week because you're studying the tissues of the body. Basically, if you're not inside a blood vessel, if you're not inside a lymphatic vessel, you're probably just chilling out in between the cells that I find everywhere in our body. And that's a space that we would call the tissues. So the fluid inside the tissues, so it's still, since we're still in an extracellular fluid, we're still outside of cells. And there can be, excuse me, there's all kinds of cells in tissues that you'll learn this week in lab. So in between any of those different types of cells, if I've got fluid there, that's called interstitial fluid. So I've got a blood vessel. We'll call it a sinusoid, a really leaky blood vessel. Some of my plasma sneaks out of that sinusoid and it goes in between the cells found in my tissues. Now the name that I call it is interstitial fluid. It's no longer called plasma because plasma is only found inside the blood vessels. Now, as I dump fluid into the tissues, I don't want that fluid to stay inside the tissues forever. I need to kind of recycle it, or I need to at least have a controlled amount of fluid that's in there. Otherwise, we're going to get swelling. Here's a correlation word for us that relates back to our water intoxication article that we read. We had a word called edema. Do any of you happen to remember what that word edema meant?
Is it swelling?
Yes, yeah, absolutely right. Edema is swelling. Swelling is what happens with too much fluid somewhere. So if I didn't have a way to get rid of my extra interstitial fluid, that would lead to a condition called edema, where I start to see swelling in a part of my body, or what we talked about in that water intoxication article, some of my cells can actually start to swell. And that's bad news when we're talking about like the cells that live inside your brain, because they have nowhere to swell to. So we need to have a way to get rid of this extra fluid that's in between my tissues. And one of the ways that I can do that is using my lymphatic system. So my lymphatic system is a lot like my circulatory system with the blood vessels in it. My lymphatic system also has vessels to it, but we call them lymphatic vessels because they're part of that system. So this is the fluid inside, I'm going to go down the line here, lymphatic vessels. And if you look in a picture, or if you do a Google search, usually for whatever reason, they've decided to make these lymphatic vessels, they look green. So they look a lot like our blood vessels do in the body, except they have a little bit different structure to their wall, and we color code them different because they're part of the lymphatic system. Elaine, you had a question about that. What's your question?
So is edema sort of like the same as when people say they have like water weight?
It can kind of be like that, yeah. If they're having an increase in their interstitial fluid, it kind of depends on where, when they talk about water weight, where they're talking about the water being. Like if it is just in between their cells, definitely that could kind of be water weight. And the amount of interstitial fluid that you have is going to change based on how hydrated you are. So yeah, that might be a good way to think about it. If we're swelling up with water, we would definitely have some water weight. So then you can say, I got to get rid of my edema, right? Instead of just saying your water weight, right? I have too much edema. I gotta deal with that. So if we want to get rid of that edema, we want to lose some of that water weight. We want to get this interstitial fluid that's inside our tissues. We want to drain it into a lymphatic vessel. The lymphatic vessels are part of the lymphatic system. That's something that you studied for lab. It's been, what, 2 weeks now, I think. Does anyone happen to remember what the lymphatic system, the function of the lymphatic system was? Or do you remember anything about the lymphatic system?
One of their jobs is like defense against pathogens.
Yes, absolutely. Yep. So here, let's make a little note for ourselves about that. The lymphatic system. In easy person words, that's what I like to call it. In easy person words, we'd call this the immune system. So the lymphatic system, like Maria mentioned for us, its biggest function is fighting off pathogens. I mentioned that to say that inside our lymphatic vessels, we're going to have those white blood cells. You'll learn a little bit about them this week in the lab packet, but really that's going to be an AMP2 topic for you there. So the big function of the lymphatic system is to use those white blood cells. They have special little tubes to travel through called your lymphatic vessels, those little green things you'll find in a textbook. They travel all throughout your body to fight off pathogens, bacteria, viruses, that kind of stuff. But to help them move, they also are surrounded by this lymph fluid. So lymph is the name of the fluid that's inside the lymphatic vessels, but it's outside white blood cells because they're moving around inside these lymphatic vessels to go to different places in the body. So what happens is we'll do our circle of life here. You've got fluid inside your red blood cells. That fluid is called plasma. Once that plasma leaves those blood vessels and leaks into the tissues of your body, so into your arms, into your legs, into your torso somewhere, I'm going to call it interstitial fluid when it's just chilling out between cells that are all over in the body. But once I have too much interstitial fluid, when it starts to kind of put pressure in the area around it, I'll actually squeeze that fluid inside my lymphatic vessels. And once it's inside the lymphatic vessels, now I call it lymph. So it's kind of three names for the same thing. Plasma, when it's inside the blood vessels, interstitial fluid, when it's just found between cells, and then that lymph fluid that I find inside the lymphatic vessels. Where it gets a little bit trippy is then my lymphatic vessels at the very end, they actually dump back into blood vessels, and then we go back to being plasma. So it just keeps recycling itself. It keeps going in a circle here. And the only difference between them, I mean, their recipes are a little bit different, right? You had that bar graph that we were learning the different types of ions or proteins, that kind of stuff. Those things are a little bit different, but the main difference between them is where I find them. So are we inside a blood vessel? Are we inside tissues? Or are we inside those lymphatic vessels? That's really how these things get their name. Where do I find you? Okay, that's the name of the fluid that's there. I think that at the beginning you already mentioned this for me, but Maria or Jalen or Elaine, help me out here. With cytosol, there's only one place that I find cytosol. I mean, I find a lot of it in this place, but what's the location of cytosol in the body? Where's cytosol found?
Inside the cells?
Yep, cytosol is inside the cells. Everything else that we've talked about was outside the red blood cells, or outside those cells found in tissues, or outside the white blood cells. My big thing to note here is that cytosol is the only kind of fluid that's found inside my cells. And so if we're talking about a red blood cell, if we're talking about the fluid that's inside the red blood cell, I call that cytosol. If we're talking about the fluid that's on the outside, we call it plasma. If we bounce over to us being inside a tissue, all the fluid that's outside those cells, now I call it interstitial fluid, but I always call that fluid inside the cell cytosol every single time without a fail. And it's the same thing with the lymphatic vessels too. If I'm inside a white blood cell, I've got cytosol in there. Otherwise, I've got that lymph fluid on the outside. So all that makes these extracellular fluids different is where they're found in the body. The only kind of fluid that I find inside a cell is that stuff called cytosol. Did that answer your question, Maria, or do you feel a little bit more clarity on those types of fluids?
Oh, yes. Yeah, yes. Thank
Perfect.
you.
Good. What else do we want to talk about? We have those fluids. We talked about our types of bonds.
I have a question about the test.
Okay, go for it.
How many questions are on the test?
I believe, let me check. I believe it is 50, but it's been a little while since I wrote it, so let me check here really fast. It's 56. There are 56 questions on the test. And some of those questions will have more than one part to them, not nearly as many parts as the homework has. Some of those questions maybe will have two parts to them, so there's kind of two blanks to fill in, or those kinds of things. But yeah, 56 questions. You have up to 75 minutes to take it. So far with your classmates who've taken it, I don't think anyone has told me they've run out of time. So should have plenty of time to answer those questions.
Okay, thank you.
Mm-hmm.
Hello, I have a question.
Okay.
In the quiz, I remember I had some question when they was asking what kind of fluid you have inside the cells. And like you do have protein for the cytosol and potassium inside the cell. But I know that you have sugar. You have a lot of sugar. And I was confused if I should the option sugar or not.
Yeah, so that's a good question about what we have in our types of fluid. It gives me a chance too to show off something. I talked about this a little bit yesterday. Were you here, Maria, yesterday when we talked about the salty banana?
Yes.
Can't
yes,
remember. Okay.
that
So the other thing too that we'll add. I think Jalen, you were here yesterday too, right? Oh, I think your microphone might have cut out on me. I know, Elaine, this is your first time, right? For the office hours. So probably salty banana is new to you.
Yeah, I caught like the last half of it yesterday.
Okay. So here we'll add to our salty banana, because if we were in class, we would do this together. When we talk about the cells of the body, think about them kind of like a salty banana. The way we described this yesterday is this is a way to help us remember the ions that I find inside versus outside of a cell. So we said that inside a cell, just like inside bananas, we got a whole lot of potassium. There's a lot of potassium in a banana. And on the outside, since this banana is a salty banana, I sprinkled some sodium on it and I sprinkled some chloride on it. But the other way that it helps us to think about it as like a banana relates to the question that Maria had for me here when she asked about glucose. Sometimes when someone has really low blood sugar, they will eat a banana to try to get their glucose level to go up, their sugar level to go up. So we would definitely say that there is a lot of glucose inside our cell or inside our banana. And I kind of joke with students, the other thing that we would say, this must be a super banana, because we're going to say that also this banana cell that we have also has a really high level of proteins. That was the other thing from that graph that we said was really high for inside the cell, or for cytosol, had a lot of proteins in it. So when you think about what I have a lot of inside versus outside, I've got a lot of potassium inside my cell. I've got a lot of proteins. And yes, we would add glucose to our list of things that there's a lot of inside. Why would a cell want to have a lot of glucose inside? What's the benefit of glucose to a cell?
Energy.
Yeah, glucose is what I use to make energy, right? So glucose is used to make energy. And our technical name for the energy that cells make, we call it ATP. That's the form of energy that they make. So I want to have all that glucose inside, because all that glucose inside is going to help me to make my energy. When we talk about proteins, there's a lot of different things that proteins help us to do, right? We talked about them kind of briefly, but can you guys help me make a list of a few of those things that we said proteins were good for? What are some of those functions of proteins?
Builder blocks from muscles.
Okay, yeah. So we can use them. So when we talk about using them in a muscle, that might fall into the category. Do you remember we talked about hemoglobin too, where it helps with the overall function of a cell. So when we're talking about, let's add a little note here. For example, with muscles, it's going to be those proteins that help with movement. That's their overall function. So in muscle cells, we're going to use proteins to help us do movement. When we're talking about red blood cells, right? I'm going to abbreviate those guys, red blood cells. That's what we said in the notes was going to help us with oxygen transport. So one of the things that proteins do for me, they help me to do kind of those big picture functions like movement or transporting oxygen. Jalen, I saw you trying to jump in there. Do you have another function that you can think of to add to our list? Oh, I wonder if I gave her trouble again. Maria, did you have another one, or Elaine? What else did we say that proteins help us to do?
It helps with enzymes.
Yeah, so proteins function as enzymes. And the job of an enzyme is it makes things happen quickly. Oh yeah, I was wondering, Jalen, about your microphone. You can always type stuff in because I can see what you're typing too. So if you want to add to our discussion that way, feel free. So when they function as enzymes, they make chemical reactions happen. So we got a whole lot of enzymes, for example, that are involved in building ATP. Or we've got a lot of enzymes that are involved in doing everything your cell does. So whether it's breaking down waste or whether it's building proteins, we use enzymes to do all those kinds of things. Absolutely. So we got proteins that help me to do my overall function. We've got proteins that help me to do very specific little things. I think there were two others. Any ideas what our other ones were? Yeah, structure, absolutely. So we called them the cells that, or the proteins that help me with structure. We called that the cytoskeleton. Cytoskeleton. So that gives the cell its structure, helps us to have our shape of the cell, which is going to be really important when we get, for example, to Unit 3, and we're talking about neurons with their really funky shapes. That is a function of those proteins as well. I think we had one more function. Am I remembering right? Or did we lump it together with something else? Was it just these three functions? Okay, yeah, so talking about what the carrier proteins do, right? So carrier proteins or even channel proteins, that gets our last function here. The last function we would say is transport. So transport across the membrane. Our last function there. So things that can't get through the membrane, those are going to be the kind of things that we would use these transport proteins for. I'll help us make a little list here. When we talk about the kinds of things that can't get across the membrane, would those things that can't get across, would we call them polar or non-polar? Who needs a protein to get inside the cell? Polar or non-polar? Yeah, polar. Polar things need a protein to get inside. If I am polar, that actually goes back to our discussion with the bonds. If I'm polar, that means that I don't share my electrons equally. So a molecule that is polar is also going to be a molecule that has dipoles because I'm not sharing my electrons. So I've got an area that's partially positive or partially negative. Or if it's not just that I have partial positives and partial negatives, if I don't just have dipoles, I could also be an ion. An ion is something that has a completely positive or completely negative charge. That's still not going to be able to sneak through the membrane. The membrane doesn't play nice with ions. And anything that is dissolved in water or likes water, anything that is hydrophilic, meaning I love water, any of those kind of things are going to have to get through the membrane using a transport protein. So when we think about those proteins that are inside our super banana here, some of those proteins are doing really big picture jobs. Some of those proteins specific chemical reactions. Some of those proteins are making my cell look the way it looks, and a lot of them too are helping me to move things across the membrane. So think about our salty banana as our super banana. It not only does it have sugar, not only does it have potassium, it's also got all those proteins in it that help it to do a lot of big important things. What else? Any other last minute thoughts or questions that we had? Let's see if I can write numbers here. I presume since you guys are here, you haven't taken the test yet, right? So we're still getting ready for the test. How do we feel about the test as we're wrapping up our studying? Membrane potential? Yeah, I could probably hit on that Jalen, and I have that video from yesterday. Yeah, so we could hit on that one more time. I have a picture of this screen, so let me pull up that graph really fast. We talked about it really in depth yesterday too, so make sure to check out that video kind of in the second half of the video from yesterday. I think help me out to know, Jalen, is it are you most interested in talking about like the names of the parts of the graph, or are you most interested in talking about the ion channels on the graph? What part would you be most interested in hearing about today? And while she's typing, I will mention, I said this yesterday and I'll mention again, I don't make you do a lot of numbers in my class, but there are a few numbers on this graph that it would be good for us to be familiar with. It's the 3 numbers that are over here on the side of the graph. The first number is what we call the resting membrane potential, which is my normal charge on the membrane of a neuron. For pretty much all the cells in my body, most of the cells in the body, I should say, because we'll talk about a difference when we talk about muscles. Most cells in my body, their normal resting membrane potential is -70. They chill out around this number to help them do their job. If I get the charge on my neuron up to -55, if I am 15 millivolts different from normal, if I hit this point, my neuron knows that I'm committed to changing my charge completely. So we talked about yesterday how this -55 number is something called threshold. Threshold is the charge that when I get to this point, all hope of staying negative is lost. I'm going to go all the way up here to +30, to the last number that we need to know here. When I'm all the way up at +30, this is the charge on my membrane when a neuron starts talking to its neighbor. So if we get all the way positive, all the way up here, this neuron is going to be able to talk to its neighbor. Anything below that, we're not talking yet. Up here at the top is where I talk to my neighbor. But once I talk to my neighbor up here at +30, I realize, oh my gosh, my charge is completely wrong. I was supposed to be -70, now I'm +30. We have a word for the charge being completely wrong, and that word is depolarized. When my neuron is all the way up here at +30, we would say that it is depolarized. It doesn't have its normal resting membrane potential. So once I get depolarized, when my charge is completely wrong, I'm going to freak out and go back down to normal. I freak out so well that I go extra normal, I go extra negative, but I'm going to end up back at resting membrane potential. So 3 important terms that match up with numbers. My resting membrane potential, that's the normal charge, -70, my neuron should chill out. -55 is what we call threshold. If I get to this charge, I know that I'm going to go all the way up to +30. I'm going to be depolarized. Since the top of my graph up here is something called depolarized, this process that I see of going from -70 all the way up to +30, I call the process we see on the left side of this graph, we call it depolarization. Meaning basically I am freaking out my membrane charge. I'm going from -70 all the way up to +30, depolarization. When I see on my other side of my graph where I'm going back down, where I'm becoming negative again, we are doing what we call repolarization. The goal is to get back to the normal charge I'm supposed to be at. Go for it, Jalen. You got a question? You can either type it or we can try to wrestle the microphone, right? I guess while she's typing, let me put this to my friends that have microphones. Do you guys happen to remember which ions moved to do these processes? So which ion helped me to depolarize, or which ion helped me to repolarize? Do we remember that from yesterday?
This question is anyone?
Yeah, for anyone. Yeah.
Okay. For depolarization, it's going to be the chemical gate sodium.
Yeah, so you're giving me a specific type here. Right now, big question was just which ion does this side of the graph. So you told me sodium. Yep. And which ion does the other side of the graph? Do you remember from yesterday?
Potassium.
Potassium. Yep, over here on the other side. These are my ions that help me do this process. Sodium, when we go back to, here's why yesterday we talked about the salty banana cell. When we talk about the process of these things moving, they can only go from where there's a lot of them to where there's a little. So when we talk about sodium rush into the cell, and because sodium has a positive charge, that's going to make my membrane charge go up. That's what we see happening here. When we talk about the movement of potassium, potassium is going to leave my cell, and it's going to take its little positive charge with it. That's what helps us to get that charge back down. So potassium leaving and sodium coming in. Yeah, and so I see your question, Jalen. There was actually a key error on that one with particular the sodium, the voltage gated sodium channels. So that is correct. You probably that point C is where they open. So let's make a little note here. I fixed it last night, so if you go back and look at your attempt again, it's going to show up as correct now. But yesterday in office hours, someone mentioned that to me, and it was keyed incorrectly. So this point C, what Jalen's question says is which point on this graph do voltage gated sodium channels first open? And the place that voltage gated sodium channels first open is right here at threshold. When I hit this negative 55 value, my charge is wrong enough that it messes up those voltage gated channels. So specifically, it's the voltage gated, since we're on this side of the graph, the voltage gated sodium channels. Now, at the very beginning, Maria mentioned this for us, the very beginning right here, these little changes in my membrane charge that get me to threshold where membrane charge is going to open up or mess up these voltage gated channels. At the very beginning here, that's going to be the work of the chemically gated channels, chemically gated sodium channels. Those are the ones that do this beginning part where things change just a little bit. If the chemically gated channels change things just a little bit, here at point C, at threshold, is where the voltage gated sodium channels will open up. And like I mentioned, that was incorrect previously. It said that I think the note saying here that it opens at D, but they do actually open here at C, is where they're first going to open. They're the channels that make this whole process happen. So at point D here, when we're doing depolarization, it is still those voltage gated sodium channels. And then we get all the way messed up. We're all the way depolarized. And at this point on the graph, this is when we switch over from being sodium doing the job to being potassium doing the job. And so this is going to be where the voltage gated potassium channels open up. I will have to check and see about where it says voltage gated potassium channels first open. It looks like maybe from the question that might be keyed wrong, so I'll check that. But where they first open is up here at the top. When we're all the way at positive 30, voltage gated potassium channels open. They're going to stay open, and they're going to be the channels that do this process of repolarization. So the entire time going down my graph over here, that is all going to be the work of voltage gated potassium channels. So they first open up here at positive. My voltage gated sodium channels first open up here at negative 55. And I will do some looking to confirm with your question, Jalen. If it flagged this as where they first open, that would be incorrect. If the question was which ones are most involved or which part of the graph shows repolarization, this is where repolarization is happening, is right here on this side of my graph coming down. But if it said that where they first open is right here, that would be a key error, and I will get that fixed right away. Yes, you're welcome. Any other specific questions that I can help you guys out with? And I'll mention, we did talk about this too yesterday. So if you want a little bit more in depth, we probably spent 15, 20 minutes on this graph yesterday. So the recording is there for that too, for a little more information.
Okay.
You're welcome, Maria. And if you guys have any questions, always feel free to reach out to me via email. If you have not already, please try to do that practice test like right now to make sure that Proctorio is working right on your computer, since that exam has to be done tonight by midnight. So for troubleshooting purposes, try to get to make sure Proctorio is all set for you. And then good luck on your exam.